the class. Welcome to Introduction to Biophotonics. Hopefully you're in the right place. Everyone in the right place?
Yeah.
Good, good. Okay, so today we're going to do the following. We're going to have introductions. We're all going to go through and introduce ourselves. And I'll have you guys sign in. I'll pass this around actually right now. It asks for your signature, your major, and whether you have a portable that you can bring to class or not. Okay? A portable computer. And if you can tell me whether it's a PC or Mac, that'd be great. Just put PC or Mac there if you have one. If you don't have one, that's fine. We won't need one for every person, but I just need to know that we have enough. Okay? We're going to then do some surveys. I'm going to tell you about the course structure. And by the way, Professor Jim Shackelford has joined us. He's the co-professor for this course. You'll be getting some examples of biophotonics, and then at around 11, we will have the director of the Center for Biophotonics, Dennis Matthews, come and talk to you, give you a broad overview of biophotonics before we dive into some of the basics starting on Wednesday. Okay? My name is Marco Molinaro, and I guess we'll start with the introductions. We're going to give our names, our major, and something fun that you did over the holidays. Okay? Or interesting. Doesn't have to be fun. Interesting. Okay? As I said, my name is Marco Molinaro. My major is biophysical chemistry. You'll notice in that word, bio, biology, physical, physics, chemistry. So when I got my PhD, I was merging different fields together. And biophotonics takes that even a step further, combining it with engineering and with medicine and other things. I think that's why this course will be particularly interesting to you. Now, what did I do that was fun over the holidays? I had the first opportunity, I have a 6-year-old and a 2-year-old, I had the first opportunity ever to go for a moonlight cross-country ski with my wife. And so on the 30th, we got our skis on, and for about an hour and a half, we went trudging through the woods, up in the mountains, of course, where there was snow, and had a really wonderful ski. It looked almost like daytime, and saw a pack of coyotes go in front of us. Kind of were wondering if they were going to come after us, but luckily they don't do that. And it was quite a beautiful, amazing time. It was actually a blue moon, what they call a blue moon, which is the second full moon within a given month. And it was a really, really nice experience, about 25 degrees, but we felt warm. Started taking off coats and stuff. So it was really, really nice. Okay.
Should I stand
Up to you. You can do it any way you like.
Stand up. Okay. I'm Thomas Bodian. I went through biochemical biology major, and I spent 64 miles over the winter.
Wow. Did it one time, or?
It was a distance camp, so we did 5 days, 5 hours a day.
5 days, 5 hours a day. And in the pool?
Yep.
Okay. Wow, wonderful. Okay. Would you like to go next?
I'm a biochemical engineering major, and I went to Tahoe with Nicole.
You went to Tahoe. Did you do something in Tahoe?
We played in the snow.
You played in the snow.
Yeah.
Okay, great. Okay.
I'm Nicole. I'm undeclared right now. And I went to Tahoe with Emma over break.
And you played in the snow.
And I also played in the snow.
Okay, great, great. Okay.
I'm Landon Ellis. I'm an economics major, and I went to the Getty Museum
The Getty Museum.
in LA.
Oh, that's beautiful.
I'm James McGeehee. I'm a biochemistry and molecular biology major, and I went and saw Avatar in 3D.
Where did you see it?
A Century.
I saw it at the IMAX. It was quite awesome.
Hi, I'm Kyle. I'm a linguistics major, and I worked at my job all break.
That was the most exciting thing that happened to you?
Oh, I also saw Avatar.
Okay.
Hey guys, I'm Bo. I'm a undeclared physical sciences major, and I went to Tahoe to snow.
Summer, wow.
My name is Lucas. I'm a biosci major, and I went back to Iowa.
Where are you from?
Huh?
So
No, it's my family's back there. So.
Okay. It was snowy, right?
Yeah.
A lot, right? They got major storms. Yeah.
I'm Christian. I'm a biomedical engineering major, and I went skiing for break.
In Tahoe or somewhere else?
Swan.
I'm Daniel. I'm a food science major, and I got to spend Christmas in Cabo San Lucas with my families.
Oh, sounds good.
My name is Becca. I'm a biochem molecular biology major, and I also went to team.
In Tahoe?
My name is Sarfaraz. I'm a psych major, and I bowled a 155 in the rig. Higher than I've ever done before, about like 70 points.
Oh, that's great.
I'm Kaylee. I'm a math and a science major for math. And the most exciting thing I did over the break was probably my pitiful attempt at snowboarding.
Hey, and you live to tell about it. So.
My name is Ben. I'm an econ major, and I went to 2 Sharks games for the race.
Nice. I'm Dominic. I'm a political science and economics major, and I went down to Phoenix for the race. The offices snow.
I'm Sam. I'm a poli-sci major, and I went shooting with all my friends.
I'm Jeff. I'm a chemical engineering major, and I went back to Orange County to see my family.
My name is Megan. I'm an international agricultural development major, and over break I went to the coast system.
Sounds fun.
Hi, I'm Sarah. I'm a political science major. I went to 1 Shark game, and I went to Christmas in the Park.
Christmas in the Park.
It's in San Jose. It's this display they put on.
Oh, that's good. Nice.
I'm Neve. I'm a chemical engineering major, and over the break, I went to a Thanks Miss Ween party.
Thanks, Miss Ween. Nice.
I'm Colin. I'm an undeclared physical science major, and for New Year's I made sushi with my family.
So I'm Matthew Slaughter, and I never left town. I'm a history major.
What are you? History major?
Yes.
I'm Rebecca. I'm an animal science major, and I decorated a photo for.
I'm Lane. I'm going to declare in economics and philosophy, and I went all over the SoCal to see my extended family.
Oh, great. Jim?
Well, I went to Orange County and walked on the beach and Laguna Beach, and I saw Avatar. Quite good.
Well, it sounds like hopefully you got to relax a little bit, got to get ready for the coming quarter. Before I talk to you about anything related to biophotonics, I need to give you some surveys. Now you might wonder, why am I giving you these surveys? There'll be 2 surveys. One is a general one about what you're hoping to get out of this course, the kinds of hopes and aspirations you have for your future. The other one is about your specific knowledge in biophotonics. Okay? Now, neither of these affect your grade in any way. They're purely for us as an aggregate. So we de-identify them, we take away all your names. In this first one I'm going to give you, you don't even have to put your name, but in the second one I give you, you do, because we have to link it back to your first exam. But we use this because we have to report to the National Science Foundation. This course is funded in part by the National Science Foundation, which is basically yours and my tax dollars. And they want reports every year, and they want to know if the students who took 10 weeks of Introduction to Biophotonics learn any biophotonics. It's a reasonable question, right? And they won't just take my word for it based on your final exam scores. They actually want to see some kind of pre-post, because you might already be biophotonics experts coming into this course. Okay? I doubt it. That's never happened before, but you never know. You might have some. So we do this pre-post kind of scenario that you'll be getting in a moment. Okay, so any questions about the surveys? These are, you do it by yourself, no teamwork on this, even though there will be a lot of things that are teamwork later in the class. This is the general one. And then after these 2 surveys, we'll begin to talk about biophotonics, the reason you're here. If you're not on that list of students that's going around, make sure to add your name at the bottom, okay? Because it looks like we have a couple of additional students. I'm passing around the syllabus for the course, just so you have it. You have about another 6 or 7 minutes to fill this thing out that I gave you. Or longer. Whose time did we inherit in this process? Anyone like more time? Okay, if you're done with that survey, why don't you pass it to the edge? Yeah, go ahead and just pass it up to the side. You can just pass it up this way. Put them all together at the edge, and I'll pick them up. Okay, I'm going to take a quick picture of you guys, and then we're going to have you write your name on Wednesday. This is just to help me remember who you are. So I'm wondering if you can move a little bit further back, just to feel like I won't be able to get you in the picture. I have a very visual memory, this is how I remember your name. Let's see. Just squeeze in this. No, just put it. Thank you. Okay, you guys ready? 1, 2. Okay, one more. 1, 2. Okay, thanks. Okay, so let me introduce to you Dr. Jody Gallacy. She's in charge of evaluation for the Center for Biophotonics. She's going to administer the more formal evaluation instrument.
Good morning. So, I'm going to give you what's called the Biophotonics Concept Inventory. And it essentially has a set of items that are sort of the core concepts in biophotonics. So this is what you would consider the pretest, right? And because you're taking a course in biophotonics, our assumption is that you probably don't know a lot about biophotonics, otherwise why be here, right? So we don't expect that you at this point will know the answer to all or many of these items. So we just want you to do the best you can. This is actually just used for our program purposes, so it has no reflection on your grade at all. What I'll do is once I collect these is we'll actually have your name removed and replace it with an identifier code. And when I report the results of this to Dr. Molinaro or to the National Science Foundation, it's reported in aggregated form as a group, and no single individual can be identified. Okay, so that's how we use it. Any questions that you have about this? Okay, so there's an answer sheet. The actual test itself is printed on color and in color and to save paper and trees and money. We don't have you write on that, just simply on the answer sheet. So I'll pass out the answer sheets, and if you just put your name and date on those, and that's where you'll record all your answers and not write them anyway on the questionnaire itself. Is there anyone who didn't get one? Are there extras?
Yeah.
So there are 14 items on this questionnaire, and you've got space for those to circle your responses to those. And then there are a few additional items that ask about sort of your interest in science in general, and then there's a question about interpreting a graph, which is an important part of science. So here's the questionnaires that you're not going to write up. doesn't have one. Okay, I'll pass that extra to you.
familiar with all of them? Anyone else still have it? Oh, I'll come over.
Thank you. I'll see you again at the end of the class.
Okay, so let's move on to the course structure. By the way, how did that test feel? Did you feel like you know all the answers? Easy? How many of you thought it was easy? How many of you thought it was hard? Everyone thought it was medium. Okay, that's a nice safe answer. Okay, so what does this course have for you? We are going to have a variety of discussions, and by discussions I really mean if you guys have questions you should interrupt, whether it's me talking, Professor Shackelford, or one of our guest speakers. We will have a variety of guest speakers that come in. Guest speakers could be graduate students, postdoctoral students, or researchers. We do have quite a nice range for you. We will have demonstrations. I'll be showing you things. You guys will get to try them. There'll be hands-on trials, and you'll actually get to observe different characteristics about light. You get to learn about them. You get to use some scientific equipment. That's why I asked you if you had the computers, because you'll be doing some actual analysis, collecting data. And you'll have discussions with scientists. The website, if you go to cbst.ucdavis.edu. You can also go to biophotonics.ucdavis.edu. But if you go to that website and you click on education, and you click on courses, you'll see that right here, winter 2010 is right there. I'll have to change this menu too so it'll be there. I haven't done that yet, sorry about that. But you click on winter 2010, and here is your syllabus. These links will become active once we have a video put up there, or the PDFs of the lectures will go up there so that you guys have access to all the materials. Any questions about that? We will have in-class quizzes. They're basically pop quizzes. You'll get about 4 of them. They're really meant to get you guys accustomed to the types of questions that you'll be seeing on the final. So there'll be some multiple choice, there'll be some short answer, and potentially some long answer, depending on how we're doing with the class. You will also be giving a presentation. You'll work in groups, and near the end of the quarter, you'll give a presentation to middle high school students, community college students, or campus students, or to the public. Or you may decide to make a movie. In the past, we've had students talk about some aspect of biophotonics to middle school students in Woodland, also in West Sacramento. We've had students go to the farmer's market and have a table on biophotonics on a Saturday morning. We've had students go into the MU and talk to other students about biophotonics and check their blood oxygen levels and their temperature and bring the infrared camera, a whole range of things that have been done. And we've also had people make a puppet show and film it. And we've had movies. We've had a cartoon, a movie cartoon that was made last year that I'll show you guys at some point. That was really good. We've had a dating game with the contestants, bachelor kind of things, asking questions about biophotonics. So quite a range. It's really your opportunity to be very creative and do something that you're really interested in doing. We will have 2 laboratory reports. So this is to give you guys a sense of what a more open-ended laboratory report done by scientists looks like. Right? Some of you will find this relatively common, familiar. You might have done this in high school. Some of you may not have had this type of experience before. The twist that we'll insert into that is that we will have a peer review cycle. So in the first laboratory that we give you, you will write up your laboratory. You will then anonymously give it to 3 other students in the class. They will give you comments on it. We'll get that back to you. So you'll get comments from the instructors and from 3 of your peers, just like we would do with a scientific journal article. And you'll get a sense of what that's like, and you'll be able to revise based on the comments, and then you get a final grade for that. In the second lab report, we won't have as much time, so we will not do a peer review cycle. And then we will have a final exam. pretty much in terms of grading, your classroom participation is 10%, quizzes are 15%, the presentations that you do or movie is 25%, laboratory reports are 25%, and the final is 25%. Okay? Any questions about that? Have you guys ever done things like the presentations? Gone out or made a movie? Or the very first year we had one group went all around Davis and just found people in the street and said, hey, what do you think biophotonics is? And recorded what they said, and it was pretty hilarious, the different things that they thought it was. So there's quite a bit of room for doing interesting things. As you'll notice, there will be a mixture of listening to someone like myself talking to you, but there will also be elements where you're doing things very actively. There'll be writing components to this course, and there will be presenting components of the course to students, but also to your class. And you'll be talking to your class about some of the things that you plan on doing with others. Okay, so let's get into biophotonics. This is a diagram you're going to see over and over again. Okay? It's really the basics of practical biophotonics. Practical biophotonics, you have a light source, some kind of light, that interacts with the subject, and that tends to be, because of biophotonics, it's something living. And then we look at how that light interacted with the subject, and we monitor the light. We try to comprehend what happened to the light with a sensor. And then that sensor gives information to some kind of interpreting device, usually some kind of computing device. It could be just a straight old computer, it could be some custom-made piece of hardware. So I'm going to give you guys some examples. Okay? And some of these you saw before, and some of these you may not have. But this is just to give you a quick preview of some of the things that you're learning more about. So, here I have a set of LEDs. Okay? Here's a blue LED. So if I stick the blue LED on my finger, what do you think you guys will see? Sorry? Nothing. Any other ideas? Hello, Dennis. Any other ideas? Okay, let's try it. So here it is, blue light. What do we see? Reddish light. Tiny little bit. Tiny little bit of light. Now I'm going to try that with green. What are we going to see with green? Same thing. Okay, let's try it. You guys see a little bit of green coming through? A little bit on the edges, where it's thinner. Huh? You see that? A little bit? Huh? Okay, now we're going to do red. What are we going to see with red? Okay, here we go. Okay, everyone sees that. Okay. Now I'm going to do one last one. I'm going to put white light. What is white light? What colors? All colors. So what will happen when I put my finger on it? You guys have probably all done this, right? Taking a flashlight, okay? You see the red comes through. Now, that red had all the colors of the rainbow in it. I mean, sorry, the white had all the colors of the rainbow in it, but only red came through. So why do we care? Why do you think that matters? That we respond to different colors of light in different ways? How might that be useful? Yes?
It can sometimes be used for just like composition of something.
Yeah. The way the different colors of light correspond to different energies of photons, right? And different energies of photons interact with the molecules inside of us in different ways. We can use that information to find out something about what's going on inside. For example, what's in my finger or your finger? What's it made of? Sorry? Blood. What else? Tissue. Bone. Sorry? Fingernail. Okay, keratin. Okay. What else? What is there a lot of in your finger? Plasma. What's plasma made up of? Did we hear water? Water. Okay, we're supposed to be 70% water, right? So there's lots of water. There's blood. What else? What other small things are in there? Platelets. Okay. How about even smaller things? Cells, even smaller. Molecules. What kinds of molecules? What families of molecules would be in your finger?
Proteins.
Organic proteins. What else?
Lipids.
Lipids, fats. Anything else?
Amino acids.
Amino acids. Keep thinking. Nucleic acids like DNA and RNA. Okay.
Ions.
Sorry?
Ions.
Ions. Yep. Yeah. Salts. Ions of salts. Yep. Anything else? Carbohydrates. Okay. Now guess what? Each of those molecules has a different way it interacts with specific energies of light. Okay? So by interacting, that light can either be absorbed, it can be reflected, it can actually cause a change in that molecule. All those different things are relevant, and we can use those in biophotonics. So, for example, you guys remember this tool? Okay? This tool takes your finger, put it on this finger, and you guys, we'll pass this around. We have to wait for it to blink green. And it tells me that his blood is 97% oxygenated, 98, and his heart rate is 70, 71. Oh, you're getting more excited here. 72. Here we go. Let's raise it. Okay. And how does it do this? How does it tell you the amount of oxygen in the blood? What do you guys see? What color of light do you see? What did we learn about red light compared to other colors of light? It tends to go through. Most of it goes through the finger. Okay? And what do you see at the other end? A little sensor, a little box. Okay? What do we talk about in practical biophotonics? What do you need? Light source, something living, detector, and interpreter. All right? This is a very, very basic example of biophotonics. Hopefully none of you have had to go to the hospital, but many people when they go to the hospital, especially elderly folks, one of the first things they'll do is they'll stick a pulse oximeter on their finger. It looks like a Band-Aid. It looks like a Band-Aid and it has a little wire, and then it plugs into a machine. And what it does is it actually figures out the amount of oxygen in your blood. It can also figure out the amount of carbon monoxide in your blood, methemoglobin, a whole range of different species, just by using different colors. Now, how do you think red light interacting? By the way, there's two lights in there. There's a red light and a near-infrared light, like a TV remote control. You know the little things that you see on your remote control when you press a button, you don't see anything light up? Have you ever noticed that? But if you look at it through a video camera or the screen of a compact camera, you'll actually see something flashing because they're sensitive to near-infrared. There's actually a red LED and a near-infrared LED, like a TV remote control in there. So how do you think they figure out the amount of oxygen in your blood? How would they do that?
That oxygen reflects light in a certain way, and that's how light is refracted.
So you think it's the interaction of the light with oxygen, right? Any other thoughts?
Or the hemoglobin is carrying it.
Hemoglobin, okay. Any other thoughts? No other, yes.
Well, more specifically what you said, we know that when blood is oxygenated, it's red, but it's greenish, or not as red, and therefore it will reflect a different color way the red light is reflecting the sun.
Excellent.
Exactly how.
So, blood with oxygen is one color, blood without oxygen is another color. What are those two colors, by the way? Sorry? Well, which one is which? So he said that red is when it's not oxygenated and purple is when it's oxygenated. Any other ideas? You think it's the other way? Okay. Any other thoughts on the colors? Are those the colors? Is it purple and red? I'm sorry to disappoint you. It's actually not purple and red. Okay. It's actually red and dark red. Okay? Now, wouldn't that be boring? You guys remember all those diagrams you saw in high school, or even if you dissect, any of you dissect animals in high school? Okay. Did they have that latex put in them that showed you the veins and the arteries? What colors were those? Pink and blue. Okay, you had pink and blue. Some of you might have had red and blue. Okay, those are usually only the colors. They do that because it's easier, right? If you put in red and dark red, you'd be like, I don't know, is this an artery or a vein? You wouldn't have any clue. So because of that difference, you've grown up with all these diagrams that have red and blue as the colors of action with of